let's move into the second part here that I'm calling Deconstruct Everything. So in this session, we're going to talk about where bias might be hiding, validation, and a few other things. This one's supposed to be the long one, so I'll try to skip over some parts that are not quite as interesting if we run out of time. So we talked about Banana Bio. I made up a completely different company for this next session, and it's called Absolute AI. And we'll say a few years ago when I first started the job, I met this company, and they said that they're defeating aging through big data. This was at the height of AI, before the AI fatigue, and AI's become a bad word in Silicon Valley now. Nobody uses it anymore, and we'll talk about why. But this is also probably the height of like anti-aging companies. I haven't actually seen that many anti-aging companies, but I feel like I heard a lot about it like 3 years ago. Maybe it was like a local maxima and there's a bigger peak coming, who knows. But this company and their pitch was really intriguing to my naive self back then, which was that they were using AI to mine a big data set to uncover a unique signature of biomarkers that achieved 99.9% accuracy in diagnosis and could also be targeted to defeat aging, right? Again, this is not a real company, but sounds very realistic to a lot of different types of companies I'm sure you've seen. Well, let's start with AI and what do you want it to mean, right? AI seems to be everything and it means nothing now, which is why nobody touts themselves as an AI company. I mean, some do and they're real, but most don't anymore. Well, I've come to think of AI as a term that vaguely means I do data things on a computer, because that's really all you can read into the term AI these days. I'm going to talk through what some people mean by AI. It could be as simple as doing regression analyses, which you could do literally by hand, and I think they were meant to be done by hand back in the day. I mean, 7th grade, use a TI-83 calculator and draw a best fit curve. That's what a regression is. But sometimes that's actually all that's needed for something like a diagnostic test or stratifying patients for different drugs. And if you do it on a computer, and usually you're talking about things that are not linear, you're talking about a multivariate regression. You have to use computers and algorithms to best fit different kind of statistical analyses to your data. And so there are still statistical regressions. I wouldn't consider this machine learning, but it does use a lot of, or it does require some computational power. But these are simple regressions, right? They're continuous and they require continuous dependent variables. If a quick refresher from algebra back from 7th grade or 8th grade or whenever that was, your dependent variables on the y and then your independent variables on the x, right? And then you're plotting your data there and then trying to figure out what a best fit curve is to that. It gets more and more complicated. The more complicated your data sets are. A quick refresher on what the R squared is. Everyone knows that an R squared of 1 is perfect, and that's what you want. That's when the dots align with the line. What it actually means in concept is the explained variation by whatever regression line that you've generated versus a total variation of the dots and how they fall away from the line. So that's kind of the conceptual explanation of how you calculate an R squared value. One thing that people may or may not be familiar with, especially if you haven't taken a statistics class, is the confidence interval, which is sometimes also shown with these regression graphs. A confidence interval, by definition, is a range of values where the true mean likely lies, which means that in the graphical form, usually you see 2 additional lines above and below any sort of regression that gives you a range in which you are confident that the true mean really lies. And what you typically see in data is a 95% confidence interval, which means you have 95% confidence that the range of values expresses where the true mean actually lies. Just to quickly make this real, it's not that complicated, right? Like if you ask me how many pennies fit in a mason jar and I say somewhere between 300 to 400, in my mind, I'm thinking there's probably a 95% chance or likelihood that those mason jars take 300 to 400 pennies. That's what a confidence interval is. And these will be important later on. So that's statistical regression, and let's talk about machine learning and what people call AI more frequently, right? Which is just machine learning. Well, one thing I wanted to point out that most machine learning is actually statistical learning. Everything still takes in data and it tries to use statistical analyses apply to the data set to do whatever they want. In this example here, categorization. So typical machine learning for categorization, you would consider taking a sample, extracting some features from that sample, running them through your algorithms, which basically go and do the statistical learning to classify your sample into disease or healthy. I'm mixing companies here because this is from Banana Bio, but just stay with me here. Basically, this is just the ability to classify and bin things. But at the end of the day, these are still statistical principles, right? So here you would think that you would call this a logistic regression instead of a linear or nonlinear regression. And here, instead of using a contiguous dependent variable, you're using a categorical dependent variable. So for example, these three types of logistic regressions, a binary logistic regression basically classifies things into two subsets, like healthy and disease. An ordinal logistic regression stratifies populations that are linked, like mild, moderate, and severe. And a nominal logistic regression separates out categories that are unrelated to each other, like three completely different disease types: lupus, Alzheimer's, cancer, etc. But these types of analyses are what the algorithms are fitting to your data sets and your features to generate these different classifications when you think about machine learning. Now, when people talk about deep learning and neural nets, the difference is that instead of manually doing feature extraction or having people do feature extraction, you're training your computer and the neural net algorithms that you've built to do everything. So you just feed in samples, and your neural net determines what's an interesting feature, and then does the classification. And here, you're removing a little bit less of what humans are doing. So computers more, humans do less, and thus there's less human bias, but maybe there's more computer bias. That's not necessarily a bad thing. Sometimes there's a bunch of features that people wouldn't see, but computers might. But that could also lead to bias. For example, in the early days of using AI for X-ray data, a typical application of deep learning to detect disease from an X-ray, they found features that doctors didn't see. And that could be good, but there could be bias. For example, these early days of AI and X-rays, the AI was, the neural nets were able to pick up differences in X-rays depending on the machine manufacturer and classified those differently. So if you're allowing your algorithms to do the feature extraction as well as the classification, that might happen, and you might say someone's disease versus healthy, but in fact it's just you have one X-ray instrument made by Hitachi versus another X-ray instrument made by GE, and that becomes your classification. Now, that doesn't really happen that frequently because most learning, machine learning, whether it's more traditional machine learning or deep learning, is supervised learning. And I'm not going to go into supervised versus unsupervised here, but in general, almost everything that you see at least in biology, seems to be supervised learning. I have yet to see unsupervised learning in any companies. So the other thing I wanted to mention is that feature extraction is built into sample prep for biology. So what do I mean by that? The reason why you don't see neural nets that frequently in a lot of life science companies that have to use some sort of machine learning to come up with different categories is because when you think about a sample, I think a sample is the patients, right? Like you have a sample of 200 disease patients. Well, usually what you're doing is taking some sort of biomarker from those patients and you're measuring that. And so when you're doing a feature extraction, that's actually built into the sample prep. You take blood from a patient, for example, you're trying to collect the DNA from that blood sample. And so you are removing everything else, the protein, the lipid, the gunk, and you just end up with a pure DNA fraction, which you feed into a sequencer, and that sequencer you feed into your algorithms for classification. In the necessity of purifying that DNA, you are by definition extracting a feature from the sample. And so because a lot of biological, life science companies and biotech companies have to rely on that sample preparation, there's a lot less that a computer can draw if you are doing all of your feature extraction into a simple biomarker. And that's why you typically see more traditional machine learning and less deep learning, where you have these neural nets in biology. That's not always true, and there's a lot of different applications, including drug screening and whatnot. But I won't go into details here. I'm sure that many of you know about those types of companies. All right, so at the end of the day, you end up with something like this, right? You end up with the categorization that's kind of good, and the more data that you feed into your algorithms, the better fit your statistical analyses can be, and you end up with a much better classification. As someone who's a trained biologist and not an AI expert, I always like to start with the output and the input, right? Because if you have great output, then it signals that there's something interesting to look at. If you have something that looks kind of crappy and the company is presenting something that looks kind of crappy, then who cares what this portion is, right? The output looks bad, so you can just stop your diligence right there. But in general, that's not the case, right? People have great output. Then I look at what the sample actually is, and we'll talk about diligencing the sample in the next few slides. But if the sample checks out and the output looks good, that means that there is some magic being worked out here in the middle. And that's when you want to dig in. As someone who's not an expert in AI, and that's just not my training, what I like to think about are the high level components of what's being done in whatever they call machine learning. So there are algorithms that are usually using statistical models, as I mentioned. So what are those? What exactly are they applying? Once you understand that, I feel like it's usually enough for you to understand how the sample got from one place to the other. When you get to neural nets, it's a completely different ballgame, and I'm just not going to go into that here because that's another can of worms. Okay, so let's talk about looking at the sample. I spent a lot of that last lecture talking about output, sensitivity, specificity, and how to dig into whether or not that's good. So let's just say that companies like Absolute AI come in and they will show you something fantastic. Great, let's move on to the input. So in the case of this company, when you ask them for specifics about the input and how they came up with something as good as this, right? They would say something like this: we trained our algorithms on 200 patient samples using over 2,000 microRNAs and 20,000 genes to feed into our proprietary algorithms. And we validated this on 2 million individuals. This is something that the company said, and you can tell that I made this up and lazily used just orders of magnitude to show that numbers can be big. And that's the point, right? A lot of times you will see large numbers and think, wow, there's a lot of data kind of going into making this. But that's not necessarily true, right? The actual data set here is the 200 patient samples. Everything else is fluff. I mean, it's important fluff, and you need to look at it, and we'll talk about that for like the next 10 minutes, but it's still fluff. The actual the sample size is 200. And so you can't really consider this to be big data, right? This is small data. This is 200. Let me give you an example of how companies use different variables and how they feed that into their algorithms. So let's start off with this phrase, right? We use over 2,000 microRNAs and 20,000 genes to feed into our proprietary algorithms. Now I know there's a lot of non-biologists here, so let me explain what a company might show you. And this is a heat map of gene expression. So this is more of like the 20,000 genes. This heat map, just to show you how to interpret it, each column is a gene and each row is a patient. And the color that you get is for gene expression, because genes are not static; they're expressed and transcribed into RNA. If I could take 30 seconds to explain the central dogma to those who might not understand it, right? You start off with DNA that gets transcribed to RNA, and then RNA gets turned into proteins, which actually do the work in biology. When someone talks about measuring, gene expression and genes in a biological sample, what they're talking about typically is RNA. And so if you have 20,000 genes, you can think of this as source code, and then the RNA is the copies of the software that are actually out there doing stuff. So for each of these 20,000 genes, there are many copies of RNA of those genes. And if there's many more copies, then that's red, and if there's less copies, then that's blue. So it's a lot of data. So if you think about 20,000 genes, that could actually be hundreds of millions of RNAs. But it seems like big data should be good, et cetera. I just mentioned it's not big data, it's big variables. But when is bigger not actually better? Well, one of the key things that you typically see is that more variables leads to overfitting. If you have a sample of 200 people and you have 20,000 variables, all which have different tuning levels of expression, then you can tweak each of those variables to fit your statistical curves to whatever you want, right? And then you end up with overfitting. We'll talk about overfitting more in a second. There's another aspect that people don't appreciate, right? I mean, who cares if it's overfit if it's perfect, and you can measure this in every patient. That's true, but there is a practical aspect as well, and this may be impractical. I love this picture from the book, The Design of Everyday Things, which you should also read. It's a fantastic book. If a company like Absolute AI comes with this data set and they say they have this crazy algorithm, chances are that they're using retrospective data, which means that somebody else collected the data and they didn't do it themselves. But if you think about practical product development and putting this in the hands of clinicians, a test in the hands of clinicians, it might be completely impractical to try to measure 20,000 genes being expressed, as well as 2,000 microRNAs and whatever else that they do. And so if you've trained your perfect algorithms on 20,000 genes and the expression of 20,000 genes, and you can't do that in clinical practice, then what's the point? Bigger is typically not better. And the other thing is, it's almost certainly biased. And this is actually a concept that you could probably imagine being around, but it's not something so simple. So let me walk through two quick examples of bias and where you might see bias in big data sets. One is with genome sequencing, right? So there's 6 billion base pairs in the human genome. I come from Illumina Ventures. Illumina Inc. makes all these sequencers. Well, sequencers are pretty accurate, and you can say that a sequencer might be 99% accurate. That still leads to what, 60 million errors. That's not a tiny amount of errors. But even so, let's assume that you have 60 million errors, and who cares? That'll kind of average out over a large sample size. Well, the problem is that the 1% error that you see is biased. It's not random. When you see this 1% error rate, it's not like you're sampling the whole genome and you're picking a random 1% every time you sample the whole genome, and that's the error. You have very specific regions in the genome in which the sequencer as a technology has a hard time handling and decoding, which means that this 1% might always be in the same regions. And if those regions happen to be important, then this 1% bias is actually quite important, despite you not really thinking about this, right? Companies don't actually present this type of stuff. You have to look into what is actually being done with the technology. And there is absolutely bias that's intrinsic to every single technique. Now, this isn't the best example because there are ways to mitigate this. And one thing is like coverage. So instead of sequencing the genome once, what you typically do in the clinical setting is sequence your genome 30 times so that you can map each sequence over each other and try to minimize the errors that way. It's not perfect, but it's still good enough. But Absolute AI is talking about measuring gene expression levels, which is what a lot of companies are doing these days, because there's a lot of data hidden in the gene expression levels. So this is a microarray. It's just one of many, many, many technologies that you can use to isolate RNA expression levels and look at how these different genes are tuned up or tuned down. But there's a lot of better tools out there. This is just an illustrative point. And in this case, let's say that there's 20,000 genes, and there's typically between 20,000 to 30,000 human genes that are known. But let's say this technology captures 30% and does not capture 70%. Seems pretty simple, right? But actually that math is not that simple, because as I mentioned before, these 20,000 genes are not static. They are producing many copies of RNAs. And again, to reiterate the first point, let's think in distributions. Each of these genes are expressing different levels of copies of RNA. And you can think about the expression of RNA as a distribution where some genes express a lot of RNA and some of them express not a lot of RNA. And it's kind of like how they say, the wealthy 1% control 60% of the wealth in this country. The top 1% of genes might actually be 60% or 70% or 80% of all the RNAs that are there. So when you're capturing this 30%, maybe you're only finding 2,000 genes in that sample. And if those 2,000 genes, which is only 10% of the genome, not 30% as this 30% capture rate might imply, if the genes that are most relevant to disease are not captured, then we have a problem. And there's actually a hidden bias within this bias, because every technique has a bias, right? And when you're saying you're capturing 30% of the RNAs, you're not randomly capturing a nice sampling of 30% of all the RNAs that are expressed. Each of these techniques that you use to capture the RNA prefer to capture certain types of RNA and not others. And so this 2,000 number might actually be 500, and you're only looking at, even if you say, we're in an unbiased way looking at 20,000 genes, you might end up only looking at 500 genes and the expression levels of 500 genes. This all sounds bad, but even then, bias might not be a deal breaker, right? As long as things are consistent. I mean, it could be a deal breaker, it could not. That X-ray example I gave in the beginning, where a deep learning algorithm picks out the different manufacturers of X-rays. One way to solve that is by looking for consistency in the bias. Basically, stick to one type of X-ray instrument. If you just use Hitachi X-rays and then use that algorithm attached only to Hitachi X-rays, then who cares? All the bias is exactly the same every time. Now you'll be able to diagnose patients that way with that. So look for consistency, and bias, you should look for it, but it might not be a deal breaker. Okay, enough about that. Let's talk about the actual sample. We've talked about the variables. What about the samples? And when are great samples not great? So absolute AI, let's say that their 200 samples were doctor annotated. And a lot of times you get samples and they're not doctor annotated. You just have, data biobanks of different samples. And you have general things like ethnicity and age and gender and whatever. But there's no doctor annotation for the disease. In this case, they had that. But that sounds good, but let's think about this, right? What if the patient population that they have are all celebrities? Absolute AI is an aging-based company, and now their sample size of these 200 patients are all from the same socioeconomic class, and they have the same X factor, which is that they don't seem to age at the same rate as the rest of the population. So if you're just looking at a sample population like this, then it's not representative of the whole population. And so even if you have the best possible data coming from this sample set, it's not going to be a good sample set because it just doesn't represent everybody else. And what if the one doctor that they used to annotate things was Dr. Oz? And I don't actually know much about this guy, all right? It's just, I know generally there's a guy named Dr. Oz who says some stuff on TV, and he might be great. But it's one doctor diagnosing the entire sample population, and every person has a bias. And is that good? Maybe it's fine, but remember, you are typically using doctor annotations to call something a true positive. So it might not be a bad idea to expand this to multiple doctors who are annotating these groups. So great data can still be garbage. Just the context is a key, just like everything else that we just talked about with bias. Let's say the absolute AI said, no, we're not actually trying to solve aging for everyone. We just care about celebrities. We're gonna, charge 50 million bucks a pop and grow $1 billion business in just, targeting celebrities and selling them diagnostics and drugs to stop, the aging process. Then actually it's a pretty good sample set, right? Because the sample is representative of the exact market that you're going after. And maybe all those celebrities would only go to Dr. Oz type to get diagnosed. So in that case, it's a great sample set. So context is key. And one thing I've learned is that entrepreneurs and the data that they collect, nobody's trying to lie. Everyone's looking for truth and trying to make a product from that truth. And you see this everywhere. I think it's true with companies and their pitches. It's true with the election we saw last month, where the polling data just didn't match with the votes again. It's a little bit better than 4 years ago, but it really wasn't that great. The experts say the exact same thing if you ask them. People typically tell the truth. It's just that if you ask the wrong people, you're not going to get a correct answer. And it's the exact same concept for samples. And you need to think about that when a company comes to you with an innovation that they've used a particular sample to generate. Okay, talked a lot about the sample. How do I know if it's real? And how do I know how do I validate what I see? So we're going to talk about retrospective and prospective data sets and validation. The first thing before I start is to mention that it's important to remember there are trade-offs, right? So absolute AI wanted to look for aging populations versus ageless. I just made that up, but let's say that's what they're looking for. They can get that data either retrospectively or prospectively. So they can create their fancy algorithms and derive their insight that they use to create their technology using retrospective data or prospective data. But you have, as a company, you have to balance the cost, the speed, and the control that you give up. Sure, there's other variables, but I think these are the big three. And investors should also keep that in mind as well, because this is important. You can't just ask a company to come up with the best possible thing, because they will always be trading off on these three things. So retrospective data is data that somebody else collected, right? So you didn't collect it, somebody else collected it. You're looking for patient samples and the types of biomarkers that you probably want to feed into your system, but somebody else collected it, and thus you have absolutely no control over what was done. You can only hope that you find what you need from what is out there. Now, the benefit is that it's cheaper and it's really fast because someone's already collected the data. You just need to pay some amount of money for it. But again, you lose the control aspect. For prospective data, you get to pick your patients, and then you can, you have to follow these patients until there's a set time that elapses or a diagnosis is reached. But you have a lot more control. The thing is, if you're collecting patients and then you're following them for a certain amount of time until they develop disease, et cetera, then you're giving up on speed. It takes time, and it costs a lot of money to do that because you have to pay for doctors and hospitals and patients and all the overhead, et cetera, to do that. So when you're developing some sort of technology, almost always what you see is that companies use retrospective data. It's kind of stupid to use prospective data to do that. But when you're validating the technology, which is different, right? Development versus validation, you can use either method. And investors always want prospective data, but when done correctly, retrospective data can be almost as good and de-risk quite a lot. So let's talk about that. When you set up a retrospective validation correctly, I mentioned that you could get similar results to a prospective data set. You can still blind your sample, you can still test enough patients, you can still have a diagnosis, you can have control groups, you can have all these things with retrospective data. The only thing, again, you give up is control of what was collected, how it was collected. Sometimes, the methods of collecting things are different between different sample types, and you need to look at lots of different sources to get enough of a sample set that you need to train your algorithms. or create your widget or what have you. But all in all, you can still set things up pretty similarly to prospective. Excuse me. So that's all great, and you can use retrospective data sets to validate that you have this technology, and then you go test it in a blinded fashion in this retrospective data set. So when people do that, there's 5 things I typically look for and evaluate. One thing is the quality of the data, right? Are they actually measuring what they needed? And if they have less control over what's collected, then how was it collected? So is the data actually good? If you're looking at RNA expression, or if you're looking for microRNAs, or if you're looking for protein, how was that collected, and is it good enough? And did they collect everything that they needed? For example, if they trained, if Absolute AI trained their technology off of 20,000 genes, but they're only collecting 15,000 genes from a retrospective data set, that's an issue. The second thing I look for is sample size, and how many people do they actually have, patients do they actually have in their sample, and how does that break down, right? I mean, I talked about the importance of making sure that your sample represents the total population, but there's also a breakdown between the control versus the disease. How many healthy people do you have versus how many diseased people do you have? Here's one example of a misleading number that I get. We trained, we had 1,000 patient retrospective data set, and we trained our algorithms off of that. Well, what they don't tell you sometimes is that 800 of those were healthy and 200 were diseased. It's not the same as having 1,000 diseased patients and an extra 1,000 healthy patients, right? I mean, that number can lie to you depending on what the breakdown is. So you have to look into what that is. Blinding is a big deal. Blinding for a prospective study, right, when you're collecting the types of patients, is quite easy because you put it in the doctor's hands to collect these patients for you, and they don't actually tell you who has what disease, nor do you know what disease they have, because sometimes you have to wait a length of time for them to develop a disease. For example, when you're doing a cancer screening diagnostic and you have a clinical trial, a prospective clinical trial, you have to wait until people get cancer. And so, by nature, prospective trials can be blinded, and you can add in additional layers of blinding. When you're talking about retrospective validation, and I have my widget that I'm testing on a retrospective data set, you have to trust that the company did the blinding properly. Otherwise, they might be picking and choosing, it might be subconscious, but they might be picking and choosing to make their data look better. So I spend a lot of time talking about what the methods look like of blinding and how they blinded their samples when they're testing and validating whatever widget they created. And then the last thing is training versus validation, which is the bane of my existence for a lot of companies that come in saying that they have retrospective validation. I'll tell you an example later, but you won't believe the number of people that think that they can train and validate on the same sample set. I mean, I understand it's hard to get retrospective samples sometimes. And let's say you can only get like 200 patients samples, and that's all you can get that have collected exactly the type of data that you need. But that doesn't mean that you can train on that sample and then take a small subset randomly selected of that same sample and say, hey, I always have between a 95 and 100% sensitivity for each of these people. Like, of course, you will have a great sensitivity number. You're validating your test on the exact same people you trained your algorithms with. Doesn't make any sense. Now, even if everything checks out here, one key thing to look out for retrospective validation is the overfitting. For some reason, even if the blinding was perfect, sample size was perfect, everything was perfectly set up, for some reason, whenever you run a prospective trial after running a retrospective trial, the numbers always decrease. I have never seen it go the other way where the numbers increase, which means that for some reason, there is some inherent bias that I'm not catching. I don't understand why it's there, but retrospective data almost always seems to give you better numbers than prospective validation. And that's something you should definitely keep an eye out for, even if everything looks perfect. All right, enough about that. Let's talk about prospective data. So, I mentioned that this is much more expensive, but you have full control over absolute everything, right? You can pick the types of people that you want. You can exclude patients that you don't want. And then you can collect all the types of biomarkers that you want with exactly the type of method that you want. And that's fantastic. You can blind things perfectly, et cetera. So how do you, but that doesn't mean prospective trials are perfect. And actually a lot of prospective validation data that I see is not perfect. So, what do I look for in prospective validation studies? I specifically look at what the trial design is. So looking at like recruitment centers, if the sample is so important and needs to be representative of the market that you're targeting, where are the patients actually coming from? Where are the recruitment centers? And who are the doctors in charge of recruiting these patients? What do the trial arms look like? How many people are in the control? Are you testing your widget in multiple different fashions? Maybe twice with one patient group versus once with one patient group. Some larger number, like for diagnostic, maybe you're including more variables for one, less variables for the other. I don't know, but what do the trial arms actually look like? Statistical power is just another fancy word for sample size. But this is a tricky one. You have to talk to someone who knows knows their biostatistics to understand if you have enough of a sample size to give you a significant value if you have a large enough effect size. So if I say that my drug, for example, decreases your tumor size by 30%, that's fine. That could be the number that comes out of the trial. But if you do not have enough patients that you've tested, then that number will not have statistical significance, and it's kind of within the noise. So are you powered? The sample size. What are the endpoints? What's actually being measured at the end? And what types of things are you measuring, as the primary endpoint? What's the main question that you're asking, as well as the secondary endpoints that you're trying to collect, additional data points that you're trying to collect to make better sense of the trial itself. And then, of course, what's the duration? Sometimes time is important. For coronavirus, timing is very important, right? Like the virus is there after a few days, and then it's there for a few weeks, and then it diminishes after a few months. And so it's hard to get, depending on when you're doing your testing, and for a trial, if you're, you can recruit patients that are at a certain, time point of symptoms. But if you are saying that my test can just detect coronavirus in any given patient at any given time, then what's the duration, and how have you tested this throughout those durations? The key thing I look for in prospective trials, just like overfitting here, is actually a statistical power. So did the company actually prove that their widget, whether it's a drug or a diagnostic or what have you, actually works? Did they actually validate it? And a lot of times this is kind of tricky. They show that there's good evidence that there's some level of validation, but at the end of the day, it was not powered enough to give you that significance. And I'm not talking about significance until the next lecture. That's kind of the last thing, but it's an important concept. As an investor, I'd say that no matter what companies do, it's actually just about stratifying risk and assigning probabilities to the risk that you're comfortable accepting. So it's always great to have a perfect prospective trial, but I know that's not possible. If a company could have that, they wouldn't be coming to me for money, right? They'd be on NASDAQ and they'd be blowing up on the public markets. So I think more about what's the level of validation that's been presented. If I ask these types of questions and look into these different attributes of any given validation set, what risk do I assign to each of these, and then what's the probability that what I'm seeing is real, and I use that to feed into an investment decision, because at the end of the day, technical diligence is just one teeny tiny portion of a full diligence. I highly recommend this book called Thinking in Bets, which I'll mention again at the end, to understand that type of mindset. It's written by a poker player. So, okay, what happened to Absolute AI? I said it's a made-up company, but there's 20 companies like Absolute AI that I could think of where we've had actual interactions with them. This is just an amalgam of all of them together. So, Absolute AI, let's kind of deviating a little bit from my previous story. Let's say that they took a retrospective data set. They extracted 20,000 microRNAs, which are different from RNAs, and they fed that into some machine learning algorithm to come up with this great test that gives you a 99.9% accuracy, which is hard to fudge, and that leads to great sensitivity specificity. That's all great, but when I asked what about being practical, they said, well, we can reduce that to 20 microRNAs that drive the largest amount of the effect size instead of looking at all 2,000. And that still gives you a sensitivity specificity of 95% and 90%, which sounded great. But once you go into the diligence, the training was the validation, which, as I mentioned, it's not great. If you look at how the microRNAs were collected, no fault of Absolute AI, is just a sample set that they were working with. There was a huge bias in the technology used to collect these microRNAs because it was an old technology. If anything, the machine learning part was something that was easy to comprehend because they were applying some very simple statistical principles. But of course, 2,000 microRNAs, they had overfitting. And when they reduce the number of variables to 20 so that there's a little bit less of overfitting, there is no validation. So I said, okay, go and validate the data however you want and then come back. So a year later, the company comes back and they've been running a prospective trial, collecting patients, feeding a machine learning algorithm, and now they have 5 microRNAs, and the results are a little bit worse, but it still looks pretty good, 90% sensitivity, 85% specificity. Well, there's something kind of wrong here, and this is actually a real example of a company that I saw many years ago that I won't mention the name of. If you're running a prospective trial, it's a little funky if you're retraining something, right? So what you want to do for validation is you want to validate that the widget you created is actually right, or it's actually true. If you are not locking your algorithms and now you're changing things and you're tweaking your algorithms again to come up with different sets of biomarkers, that means you're not actually validating anything that you developed before, right? You're just developing a new thing. And this was one thing that I don't think the company quite understood. They were running a prospective trial, but they were tweaking their algorithm to generate other sets of microRNAs. In fact, the 5 microRNAs mentioned here, were only 3 of them were mentioned in the subset of 20 that they mentioned to me a year ago, and 2 of them were brand new. So what are they validating? Absolutely nothing, right? This is something brand new, and then they would have to go and validate this thing again. And so, suffice it to say, we didn't invest, and I can think of at least 2 different examples of companies where, unfortunately, I've heard of the lead investor offering them term sheets and pulling them. We've never had to actually do that, but that can happen if you're not careful about how you're validating data. That also shows you that investors aren't always that careful in looking for validation before they offer term sheets. So think about that. All right, the last concept I want to mention here, I know this is a little bit long, is this idea of correlation and causation, right? This famous Latin phrase, post hoc ergo propter hoc. After this, therefore, because of this. Well, the second part of what Absolute AI said, outside of the diagnostic portion, is that they can actually target these to defeat aging. So if we assume something like that to be true, let's say that the company took a retrospective data set, looked at the entire transcriptome or all the 20,000 gene expression levels, fed that into an algorithm and came up with 5 genes that were able to divide patients between the aging and the ageless. That sounds intriguing, right? And theoretically, you can think that, yes, if you target these genes, maybe you can actually change the ones who are aging and turn them into an ageless population. So is that actually true? Well, everyone's favorite answer, maybe, but probably not. This is where you really want to turn to literature. I've seen this example, in lots of different companies where they do this and they come up with some new biology and it stratifies patients beautifully, and they say we're going to make drugs that target these particular genes or proteins or whatever they found, and that's going to reverse the disease. And it could be true, but when you dig into literature and what's known, maybe 3 of these 5 genes actually make sense and are implicated in the disease progression for whatever disease they want to cure. But then 2 of them are completely out of nowhere. And at that point, the question is, are they actually relevant or are they causative of the disease? Maybe they just happen to be correlated with patients that have this disease, and it's great for a diagnostic, but it's not great as a drug. And you have to turn to literature and looking at journal articles to see what the scientific community thinks about these types of biomarkers that are developed, to see and assign some probability to whether or not something's correlative or causative. If you're an early stage investor, you might accept a higher risk if you feel like it's new biology, but there's some signal that it's implicated in disease, versus if you're a late stage investor, you probably don't want to take that type of risk. This is relevant in today's world. I just want to mention this one example. There's a drug called aducanumab by this company called Biogen, and aducanumab targets a protein called beta amyloid. I'm sure a lot of you on this call have heard of beta amyloid before. It is one of the key components of plaque buildup in your brain that causes dementia and Alzheimer's. I say causes, but it's not actually, that term causes, it might just be correlative. Well, a lot of companies have tried to target beta amyloid before, but most of them stop beta amyloid from accumulating in the brain and creating more plaque. Biogen, I think, had the only drug that was shown to remove beta amyloid from existing plaque, so that you're now reducing the amount of plaque in the brain. Well, they ran this crazy phase 3 clinical trial to try to get their drug approved, thousands and thousands of patients, and something like a year and a half ago, the results came back and there was absolutely no effect between their drug and the control arm, which goes to show beta amyloid's been around for a very long time. People have been studying Alzheimer's for a very long time. And if you look at the literature and you talk to neuroscientists, there is still not consensus on whether or not beta amyloid is causative of dementia and Alzheimer's versus a hallmark of Alzheimer's and dementia. So maybe by the time you have a lot of plaque buildup and beta amyloid buildup in your brain, it just shows that your brain is so damaged that you have Alzheimer's disease, but maybe the plaque buildup itself or the beta amyloid buildup itself is not causing that brain damage. This whole causation versus correlation is actually quite hard to figure out. And for something like this that's been studied for like 4 decades, the fact that people still don't know the answer, and if anything, companies have spent hundreds and hundreds, if not billions of dollars trying to target beta amyloid and have all failed. It shows you that correlation versus causation is a real thing. If you care about statistics, look into what's happening with Biogen right now. Actually, what they did is take that clinical trial, and they are now trying to do some statistical shenanigans, looking at a very small section of patients that did seem to show response, and using that data set to get their drug approved through the FDA without running an extra clinical trial. It's this big scandal. I'll call this scandal. Maybe it's not a scandal. I don't know. But there's a lot of debate on whether or not that's the right thing to do. And a lot of it is just statistical shenanigans. Okay, I've talked a lot. That session went on for quite a while, so I'm going to stop here. It's just a fun cartoon talking about samples and what people come back with for samples typically. So let's take a break.